I work as a postdoc at Drexel University in Philadelphia, and my lab, we get movies from biologists of various cells from microscopes that can take time-lapse movies of them, and our software can segment and track the cells. That's what's going on the left, and also create a lineage tree, which is being shown on the right. So like a family tree of however all the cells are dividing. These are neural stem cells from a mouse. I'll be showing you other types of cells later on. And so we end up making lots of movies as part of our work, both because they look really nice, good at presentations, and also to help visualize what's going on. And I'm running this separately because I couldn't get it to run inside of Keynote. It kept having problems. So now let's go back over here. Right, as you'll see, this is going to stop. Right, great. Yeah. I don't know why that wasn't working. This is another movie that I made. On the left, I'm trying to compare how I was segmenting that blob right there. And so the old way, it wasn't working very well, but I made some changes, and I'm kind of drawing a nice red circle around most of the places. So this is another example of like how we make movies, and I want to show things side by side, and I want to talk about how I do that. So what we normally get from our biologists are lots and lots of pictures that look. This is maybe one of the not so great ones, at least from our point of view, as trying to analyze these images. So these are hematopoietic stem cells. So they're like blood immune system cells. And in fact, we weren't even sure to start off with what we were really looking for here. So our collaborators sent us back, and they said that these really dark things, which you probably can't even see in the back, are dead cells. The sort of brighter white ones are live cells. So we're basically trying to find those little white circles in all of the movies. What I did here was just make a movie of the first 100 frames to kind of get a better sense of what was going on with these. Normally we use a program on Windows called Earthfan View, which is really good for scanning through pictures, but I have a Mac and it doesn't run on the Mac. So you can kind of see that this oddly shaped thing around the side, that's actually the well that they were growing them in. I still don't really understand why they all look so odd, and they're all different from every single one of the movies that we got. But you kind of see, it's a little hard to see probably, is one cell division that's right there. And the cells are kind of moving around. Even the dead ones are moving around, and I don't understand that either. So this is a summary of what we're looking at for this particular set of experiments. They had 52 movies, and each of them had about like 3,500 frames. There were also fluorescent frames, which are separate. They took phase, so this is a phase contrast image on the right, which is a special way of doing microscopy that enhances contrast, similar to just regular visual microscopes. At any rate, that works out to about, they took phase every 2 minutes and fluorescence every hour, except when they took a fluorescent image, there wasn't a phase image, and it's complicated. So it's about 5 days worth of data. Each of the images, it says about 1388 by 1040 TIFF, so it's about 2 megs each. And so it works out, there's a lot of data in one of these experiments to try to get a handle on. So this is kind of the basic workflow, at least for, this is not what we normally do, because this is my workflow for just sort of visualizing what we had before we'd even try to get it into our program. I'm going to go through everything except the first, although unfortunately the first is the only one that I really use Perl for, because when we get the data from the biologist out of the microscope software, it's often in very odd naming formats. Perl's great for taking a ton of directories of files and renaming them into something that's more standard. So then we're going to remove the background from these wells and then try to do the segmentation and make some montages and some movies after that. So background removal. So here's that 100-frame movie again. And if we want to segment it, this is going to be really difficult for a number of reasons. One is that the basic thing is that the outline of it is going to be a real kind of pain in the butt. It's not even like a really, because they're different in every one of these movies, and they're kind of these random curved shapes. So you can't really use like a normal, you can't look for lines or curves very easily. But what you can do is look for the pixels that don't move. So you can tell that there's all those pixels in the middle, or the cells are moving around, but the outline is pretty stationary. And a lot of the kind of junk in there is also pretty stationary. So one way to do this is you can make a stack of images. I took the first 1,000 frames, and then I took the mean in the z direction. So then what that means is that the cells that are in this wall, they're not moving at all. So their mean in the pixels on the walls of this, they're not moving at all. So they're going to be pretty high when you take the mean in the time direction. But the ones in the middle, where the cells might only pass through them, some small percentage of the time, they're going to be pretty dark. So then you can use that as a mask, and then you can subtract that off of each one of the images. So this is the original image that I showed you before. This is that temporal mask taking the first 1,000 frames and then taking the average in the time direction. And then if you subtract it off, that's pretty good, right? So that's almost the cells. Some junk, but we can deal with the junk later on. So now we want to try to find where all the cells are. And there's lots and lots of different ways to do this. One that worked pretty well for these is something called image morphology. In particular, this thing at the bottom called morphological image gradient. So what we ended up doing, the way this works is that there's two kind of core functions you can use, erosion and dilation. Erosion says that for every pixel, you look at a small circle of pixels around it, and you basically erase some small percentage, some little circle of the cells around each, some circle of the pixels around each one of the pixels that's there. But you keep the pixel it is there, you just sort of erase a bunch of its neighbors. So what you end up doing, it's like taking, I was going to say it's like taking something in a drawing program, although this is more like that. So let me get back to that. So dilation is the opposite. It takes every one of the pixels that's lit up and takes a small circle around it and lights those up as well. So that's like that's using a fat brush in a drawing program to make everything a little bit fatter. And erosion is making everything a little bit thinner. So then the trick is that if you take two images, one where everything's dilated, made a little bit fatter, and another where you erode things, you make everything a little bit thinner, and you subtract the two, you get a good estimate of what the outline of all the objects in the scene are, which is shown here. And these images are all from OpenCV Python tutorials, because there's a pretty good support for OpenCV in Python. So in practice, this doesn't always work for cells, because often they're not evenly lit. They might be brighter on one side than on the other because of how they've been lit up, or just weirdness in how phase contrast works. But it worked okay for this. So this is our image after we subtracted the mask off. This is what we get after we do the morphological image gradient. So you can see that all the cells are lit up pretty well now, but unfortunately we have some of that mask, the wall back, that we thought we had erased in that previous step. But we can still deal with that. So the next thing to do after that, is to run something called Atsu thresholding. So what Atsu thresholding does is it tries to separate the foreground from the background. And what it does is it tries to do that such that the variance is between the cells in the foreground, all the pixels in the foreground and all the pixels in the background is maximized. It's like a one-line command in MATLAB to do that. So then you end up with this, and so we have all the cells, but we also have a lot of that wall still that's there. So what I did then was just do another erosion after that to get rid of some of the chunk. And this is something you actually work with pretty well. So what you do after that is, this sort of summarizes it, and I'll go through the final couple of steps. So find something to try to enhance the contrast of wherever the cells happen to be. And morphological image gradient worked okay for this. And then Atsu threshold, and try to have some sort of foreground and background thing. Then you want to find the connected components. So all the areas that are connected. This is like a normal computer sciencey thing. So like that's a connected component. That is this long thing going up here is. And then once you have the connected components, you can maybe look at, if you want to remove things that look like the outlines, you can look at how big they are, how many pixels they are, their dimensions, so the length versus their width, things like that to kind of narrow things down. And then finally, we find the convex hulls of the cells. So which is, a convex hull is just the, it's like if you had taken a rubber band and wrapped it around the cells. All right. So, okay, now we want to make the movie frames. So what I wanted to end up with is something that looked like this, with 6 frames in it. The one on the upper left, that's the original frame. This is the one after I've found the convex hulls and all the cells, gone through all those steps I just described, with these little red circles around them. And these are the 4 fluorescent images. And I really just wanted to see what our data looked like. That was what I was going through here. So, yeah, I forgot that I actually said this. So to do this, we have to make a multi-panel image. And it turns out this is fairly easy to do in MATLAB, although I think probably not as so easy to do in Perl. So the way you can do this is that the images are just matrices. And generally, we have them so that the pixel values are all between 0 and 1, where 0 would be black and 1 would be white. So then these white lines, like right here, this is just a bunch of 1s. This is just a column of 1s going all the way up. And this white line here is just a row of 1s going all the way across. And look, MATLAB's really easy at constructing matrices. It's really good at constructing matrices. Just like Perl's really good at dealing with strings, MATLAB's really good at dealing with that. So if you're not familiar with MATLAB, can I get my cursor over there? That would be really nice. So this is how you make a simple matrix in MATLAB. You say, put them, all the matrices in square brackets. And if you set the colon, the entries are separated by spaces and rows by semicolons. So this just makes a matrix with 1, 2, 3, 4. And then B is 5, 6, 7, 8. And you use the same syntax. If they're matrices instead of just numbers, constants, it works the same way. So if you say A, B, that's just going to concatenate them going across. So this is A and this is B. And you can also put them on top of each other. And if A, B, C, and D are images like they were on that previous slide, it works exactly the same way. And it's very easy to make these montages just by doing that in MATLAB. And in Python and NumPy too, it's very easy to do. So when we make our movies, we try to make all the frames be 1080p dimensions. So in that case, there's 6 panels, and if you do the math, every one of those panels has to work out to be 539 by 638. And I try to, it doesn't always work out that way, but I made the horizontal line be 2 pixels high and the vertical line be 3 pixels wide, and that just made everything work out evenly. There's also, I don't know if you can see here, but there's some text. There's some tiny text, like right there. I probably should have made the text bigger in this. The text is always problematic. In MATLAB, what you do, there's a text function, but that always has to be inside a figure. So you have to take your image. 5 minutes left? Okay, good. So you have to put the image into a figure, use the text command to put text on there, and then pull the image out of the figure back into a matrix, and then save it off somewhere else. It would be really nice if you could use that for, there's no way to just do it totally in memory. So if you had something like in that first movie that I showed you, where that thing would be 1080p all the way up, you need to have the figure really be big. Your monitor has to be bigger than 1080p, because otherwise MATLAB will resize your window, and it's just a pain in the butt. There's some other alternatives. Pillow, which is part of the Python image library, although strangely not part of NumPy as far as I could tell. Seem to do a pretty good job of annotating frames as well. Someone was telling me that FFmpeg might be able to do it directly as well. Okay, so this is what the final movie is. It going? When did that go? We're not playing. Okay, I don't know why that's not playing. Oh, right. So then to make the movie, I write each one of the frames out to a separate directory. And it's very important. It's really helpful to write every frame out individually. I mean, MATLAB can make the movies directly, but it's a lot more flexible if you can write all the frames out separately and then make the movie after that. So you put them all in a directory, name, number things so that they're sequential. Then you use FFmpeg to make a movie after that. And so this is what the movie ended up looking like. The fluorescent things look a little weird because they're only updating every 30 frames. You can also see that the contrast is really odd. That happens a lot. I don't understand why. Like why some of the fluorescent frames have different contrasts than other ones. So then what we really wanted to look at was lots of, like, get a better sense of what all these experiments look like. So I took all those frames that I made to do one movie and then combined them all together. This is showing 16 of them all in one giant montage. And I could do that because I wrote out all the frames for that montage movie I made, so I could read the montage things back in and make another movie out of all the individual ones. I even made a 4K version of this, and all I had to do was just resize things differently, and it all just kind of worked. For some more information, that's my lab's website, bioimage.coe.drexel.edu. And that Gonzalez and Woods book is really good. It uses MATLAB, but most of the concepts are kind of general image analysis concepts and not just things that you can do in MATLAB. So, thank you. Any questions? Yeah. Separate what frames? Fluorescent frames. It depends. So the question is, why did they have the separate fluorescent frames? And it really varies by experiment. Those would have been 4 different colors of fluorescent. I think, let me back up. I think it was violet, green. Yeah, they're trying to point out different things. They can get triggered when different things happen. And they're telling different things to the biologists. So they're not really, we didn't get to a point yet with this where they're telling anything to us. Yes?
I noticed that there's a lot of jitter in the images.
Yeah, there is a lot of jitter in the images.
Is that something that you would correct first? Will that make the other steps easier?
Yeah, as long as it's not too bad, it doesn't throw us off because the cells themselves are moving anyway. So it can throw us off if it's a lot, but the amount that they're jittering here, that's not enough to really throw it off. We start having problems when if the jitter was more than like the width of a cell, which I don't think.
Does that affect your ability to identify the background?
No, it would be more of a problem in trying to track the cells, which I didn't talk about at all here. Anything else? Okay, well, thank you.